The dissertation proposal defense of Julian Egger, who is a PhD candidate in the bioinformatics and computational biomedicine major. And as we always do, the candidate defends their proposal, and if acceptable to the committee and the rest of the audience doesn't uncover any major problems, they then advance to do their dissertation work, and then we'll come back some number of months down the road to defend their dissertation. Julian received his bachelor's degree in bioinformatics with a minor in computer science from the University of Nebraska, Omaha in 2014. And he has had a variety of other jobs working as a bioinformatics programmer at the Omaha Henry Doorly Zoo Genetics Department. He's also spent some time in industry as a software engineer at Thermo Fisher, and his research interest is focused on the study of genomics using RNA sequencing with an emphasis on alternative splicing in cellular differentiation and disease. So we will let him get going.
All right. Okay, thank you, Bill. Thank you, everyone, for coming to my proposal. So the title of my proposal is Alternative Splicing Event Signatures of Drug Response in AML. So just a quick overview of what I'll be talking about. I'll give a brief background on what AML is and what alternative splicing is. And then as I go through my specific aims, you'll hear me discuss kind of an overarching theme of my project, which is centered around a methodology known as event-based splicing methods. And so what is acute myeloid leukemia, or AML? Acute AML is a cancer of myeloid blood cells in which we have these abnormal myoblasts that would normally differentiate into different types of white blood cells begin to accumulate in the blood and bone marrow. And as these abnormal cells start to accumulate, that leads to a loss of other types of blood cells like red blood cells and platelet cells. And that leads to some of the known symptoms of AML, including inability to fight infection, fatigue and loss of breath, and increased bruising and bleeding. And so AML, there's about approximately 20,000 people are diagnosed in the United States annually with AML, and around 10,000 annual deaths in the U.S. It's characterized by a high rate of patient relapse after clinical remission, and it has a very low 5-year survival rate that, as you can see, significantly decreases with patient age. So how does AML develop? AML develops through different genetic changes in the hematopoietic stem and progenitor stem cell lineage. So if we look at normal hematopoiesis, we have our hematopoietic stem cells, which can either go down the lineage and become lymphoid cells, or they can go down an alternate lineage and become myeloid cells. In the case of AML, we have different genetic changes that occur somewhere around the stem cell area of the lineage or in the progenitor cells. And as these genetic events occur, this leads to these cells becoming leukemic cells, which either the leukemic stem cells or the leukemic progenitor cells, and it's these abnormal myeloid leukemic cells that begin to accumulate. One note about AML is that this genetic development that leads to these abnormal cells is a much more rapid development compared to that of chronic myeloid leukemia. So these different genetic events that I'm talking about, these can be cytogenetic events like different chromosome abnormalities like translocations and inversions, or they can be small genetic events like mutations, so missense mutations or like indels. And even further, the presence of these different genetic events have significant prognostic implications. So for instance, this is the European Leukemia Net Prognostic System, where it shows that the presence of these different cytogenetic events or somatic gene mutations can be used to assess the overall risk of the different patients and put them into different risk categories as either favorable, intermediate, or adverse. And additionally, new mutational events still continue to be identified in AML patients, and many of these show prognostic implications. And this has led to the development of new targeted therapies to address these different types of mutations. However, a challenge of AML therapy development is that the mutational landscape is highly heterogeneous and complex, where patients can harbor a variety of different mutations and combinations across thousands of genes. And the presence of these different combinations can lead to a varying degree in drug response across the different AML patients. In addition to the cytogenetic events and gene mutations that I discussed earlier, It's also known that there are different alternative splicing events that are significant in AML. For instance, there's splicing events that are found that are differentiated between normal patients and AML patients. Some of these are recurrent splicing events that are seen through at least a significant portion of AML patients, but just like the different cytogenetic and gene mutation events, it's a very heterogeneous landscape where patients can harbor different combinations of aberrant splicing events. And like I said, a lot of these have different prognostic implications, including aggressiveness. They can be associated with drug resistance, relapse, and survival. So what is alternative splicing? So in RNA splicing, during gene expression, we have a pre-mRNA transcripts, and in this pre-mRNA, we have certain regions known as introns, which are removed, and the remaining regions known as exons are left in, and these are spliced together to form the final mature mRNA product. And then this mRNA is eventually translated into a functional protein. So with alternative splicing, we can actually use different combinations of these exonic regions, and this allows a single gene to produce multiple mature mRNA transcripts, and these mature mRNA transcripts could potentially translate into fully functional proteins with differing functionalities between them, all from a single gene. And so there's kind of different ways to categorize the different types of splicing. So we do this in the form of alternative splicing events, which focuses on the patterns of splicing. So for instance, we can have just a simple exon skipping event where one transcript will include a particular exon, another transcript, an alternative transcript would skip the exon or not include that exon. But we can have other types of events as well, where we can have we can use or partially use different exons. So we're going to have alternative splice sites where we use all of the exon, or we only use part of the exon. But we also have a special case of exon skipping events where the exons are skipped in a mutually exclusive manner, where one transcript will only contain one or the other exon. We can also have intron retention events where actually the entire intronic region that would normally be spliced out is completely retained within the final mRNA transcript. And we can also categorize these splicing events in terms of the alternative, which is the first exon used and which is the last exon used in our transcript. But we can also have go beyond just basic splicing events, and this will kind of be significant as I go through my talk, where we can have skipping of actual multiple exons at once. So this is known as exon cluster skipping, essentially. And we can also have compound events where we might have more than one basic event compounded into a single event. For instance, we could have a particular event could include or exclude skipping while also including or excluding different 5' or 3' splice sites. It's also been shown that some of these splicing events, they're tightly regulated in normal biology, but when they're deregulated, they can lead to different disease and cancer. And specifically in the case of cancer, it's been shown that the expression of these different splicing events can have an impact on drug resistance. So for instance, this BIM gene, we have this exon skipping event. In normal biology, both of these events are typically expressed in a regulated manner, but it's been shown that if there's an increase in the expression of the skipped exon version of the splice variant, that increase in expression is associated with resistance to tyrosine kinase inhibitors. In this case, this is an example in chronic myeloid leukemia. And so this leads me to my central hypothesis for my project, which is that therapeutic resistance in AML can be partially characterized by the expression of distinct splice variants, and transcriptome-wide coordination of splicing events influences response to treatment. And so before I get into my specific aims, I'd like to talk about the data set that I'll be using. And so the data I'm using is actually derived from the BEAT AML program, which is a collaborative research program led here by OHSU that involves multiple universities and multiple pharmaceutical companies. And it's actually the largest to date AML patient data set. And what's so significant about this data set is that it involves all different kinds of data. It not only has clinical data about the patients, but it also includes cytogenetic data, mutation data from whole exome sequencing, expression data, so we can look at gene expression from RNA sequencing, and it also has drug response for multiple drug types. And just to kind of summarize actually just last year, they published a paper as sort of a first insight of looking into the functional genomic landscape of this data. And just to kind of summarize some of the key things that they did is they were looking at associations of mutation status and gene expression and looking at the associations of those genetic events in terms of drug response. So this is a Circos plot that shows different mutation, all these different genetic events across the entire transcriptome, labeled by different chromosome, labeled by chromosome. In the inner part of the circle, we can see the different AML chromosome rearrangements. And then this first inner circle here, each dot represents a particular mutation that was found to be highly associated with drug response for any of these different inhibitors. And then the next circle out, these are genes. So this represents genes that were significantly differently expressed between resistant and sensitive patients. So looking more at the data set from this that I'll be using the RNA sequencing data as well as the drug response values. And so 411 of patients of that cohort have RNA sequencing samples derived from either peripheral blood or bone marrow tissue. This is just a hierarchical clustering and a heat map showing the most variable expressed genes. So this is gene expression. This is expression at the gene level. I'll be doing my project at the splicing level, and I'll discuss how I'm doing that here in a minute. And then I'll be using the drug response values, like I said. So this is a hierarchical clustering of the different drugs for all the different inhibitors across the patients. And so my first specific aim is to identify splicing event expression signatures of drug response in AML using a random forest model. So what's a random forest? Random forest is a supervised learning algorithm, and it's actually an ensemble method in that it involves a collection of decision trees in which each tree is actually given a vote in predicting the outcome for a particular observation. And so to build a random forest, we build a decision tree one at a time. In this particular example, this is a building of a decision tree. This is sort of an arbitrary example where we're predicting relapse and non-relapse using gene expression data. And so when we build each tree, what we do is we take a random subsample of our observations, and then we leave an out-of-bag sample to assess the accuracy of each tree. And as we build each tree, what we do is we take our training sample, and we also take, I forgot to mention that we take a random subsample of all of our features as well, and we choose from that random subsample of features the best feature that can separate our observations into one of our two possible outcomes. And we do this iteratively through so many iterations until a certain point where we keep choosing the feature that best separates the observation. So in this case, non-relapse and relapse, and we keep doing that to build each tree. And then after we've done this for, for example, 10,000 trees, we build our forest, and then like I mentioned, each tree in our forest gets a vote in predicting the overall outcome. And so an important point, and this is kind of why I'm looking at utilizing a random forest, is that a random forest actually provides a variable importance measure. And so for each tree that we build, if we see variables or features that were frequently chosen over others, the more frequently chosen features will be given a higher variable importance. And so this is one thing I'm going to use for addressing this specific aim. So just kind of a use case example, this is a paper where they're looking at resistance and sensitivity in multiple myeloma, where they used a random forest approach to identify a 42 gene expression signature of drug response. And so this is just a heat map of showing the signature that they produced from the random forest procedure. And this just shows the genes that represent that gene expression signature, and then it shows how the genes are upregulated and downregulated in either sensitive or resistance. For this is a proteome inhibitor. So how do I do this at the splicing level? So there's different ways to quantify alternative splicing, and I'll be using this event-based approach where we focus on these localized alternative splicing events that I mentioned earlier. And what we do is we represent the expression as the relative inclusion of each event, and I'll discuss this here a little bit more in a second. And this allows us to quantify alternative splicing without the complexity issues of full-length transcript assembly and estimation, which is a limitation of short-read RNA sequencing. So how do we do this? So what we do is we isolate patterns of splicing from either gene annotations or read alignments. So right here, we're looking at, this is a single gene in which we can have 4 alternatively spliced transcripts, each with their own sort of set of exon structures. Here we also have a reference genome and our aligned reads from a particular data set. And so we isolate out the different events. In this case, this is a skipped exon event that was found in our annotation, so this is a known event. And then we collapse that event down into this sort of single unit that represents the binary outcome of a possible splicing event. And then we quantify it using our reads, and we quantify it using a metric known as percent spliced in, which represents the relative inclusion of one event, in this case the skipped exon, the inclusion of the skipped exon, over the total expression of both possible outcomes. And so this allows us to quantify splicing without having to try and quantify the expression of all these individual transcripts, which is an issue considering our read size and the fact that many of these reads could actually belong to multiple transcripts because they share all or part of the exons. And so kind of an overall look of the specific aim. So for each drug, I'll first filter splicing events using some sort of pre-filtering method. For instance, I could filter away the least variable splicing events in terms of their PSI. And then from those filtered events, I would build a random forest model predicting drug response. So for instance, I could use a regression forest that predicts the drug response variable, which is represented as the area under the curve from the ex vivo drug response assay. And then from there, using the variable importance, I would identify significant splicing events as the signatures for each drug. An alternative approach, though, that I'm looking at using is actually a classification forest, and this is kind of based off what they did in the Beat AML paper. They took the top 20 and the top and bottom 20% in terms of drug response and sort of pre-labeled those as either sensitive or resistant. And in that case, what I would do is I would pre-label those and actually build a classification for us for predicting either sensitivity or resistance. And in the same effort, I would then find my significant splicing events to represent each drug. Some considerations, though. For one, this will certainly be done through a cross-validation framework. Another thing is the pre-filtering of events. I mentioned one possibility I could do is looking at variance. Another thing I could do also, like in the case of the classification model, if the variance doesn't filter the events down enough, keep in mind that for each gene, we could actually have multiple events, maybe dozens of events. So there could be thousands of events that we'd be putting into our model. We could pre-filter based on some sort of differential expression cutoff. In this case, it'd be differential splicing, where we could include events that have, for instance, a change in PSI of maybe 5%. Another consideration is sample size. So I mentioned that there was 363 patient samples that have RNA-seq and drug response for these 122 inhibitors. However, not every patient has a drug response for every inhibitor. So for some of the drugs, we may actually have too small of a sample size to do something like stratifying the patient samples into either sensitivity or resistance. But the expected outcome, as I mentioned, is a splicing event expression profile for each drug, where, for instance, we might have for a particular drug, there may be a particular skipped exon, where the inclusion of that exon, the expression of the included version is upregulated in the sensitivity patients, and then the exclusion version is upregulated in the resistant patients. And it's also important to point out that the outcome of this aim will give us a set of independent splicing events. So if we look more into transcriptional regulation, it's actually, like I mentioned, it's a highly coordinated process, but even further, gene expression changes or splice variant expression changes, they don't occur independently. We tend to have multiple genes operating and interacting within cellular pathways. So an example of this is the cellular differentiation program known as epithelial mesenchymal transition, or EMT, in which these epithelial cells go through a transition process and become these mesenchymal cells with a different phenotype. And this transition is driven by coordinated changes of different genes. So we have dozens of genes that are upregulated and downregulated in a coordinated fashion to drive EMT. And so a way that we can study this sort of coordination of regulation is using gene co-expression networks, where we can study gene expression now on a systems level and not just look at differential expression at the individual gene level. And so gene co-expression networks, they allow us to characterize co-expression patterns between genes, and we can study how and with whom that the genes interact. We can also identify clusters or neighborhoods of genes that are driving a particular phenotype, and by doing that, we can look at the biological functions and the pathways that those genes may be involved in. It's important to point out that in order to do this, we're using co-expression networks under a key assumption, and that groups of highly correlated genes cooperate within related pathways and functions. So bringing up the EMT example again, not only is EMT driven by coordinated changes in gene expression, but it's also driven by coordinated changes in the expression of distinct splice variants. So how can we study coordinated splicing on a systems level? And so there's been different approaches to look at a network view of splicing, one of which is a transcript co-expression network where our nodes are transcripts and we look at the correlation of the transcript expression across samples. In order to do this, though, we have to assemble and estimate the abundance of full-length transcripts. And this is kind of an issue with short reads because we have these certain reads that are ambiguous in terms of which transcript that it originated from. And so this means that when we have our expression for each transcript, we're using a statistical model to estimate that expression. And so a key limitation in using a transcript network is we could have an incorrect assembly step, and we could also have incorrect estimations for the expression of those transcripts. So a way that we can alleviate that complexity is we could do an exon co-expression network, where now our nodes are exons, and we're looking at the correlation of exon expression across samples. And to do this, what we do is we create a unionized set of exons, where we create this sort of overlapping set from all of our exon structures from all of our transcripts, and then we quantify the expression of those exons. We build a network measuring the correlation of the exon expression. However, an alternative approach to doing this is rather than quantifying the total expression of each exon, we could quantify the relative expression of each exon and now measure the correlations of the relative exon expression, where it's the exon expression relative to the total expression of the gene. And so if we do this, we can see that this starts to resemble that of like an exon skipping network because we're doing relative inclusion. And so an example of this was this paper by Sebastian et al. Last year where they did an exon network of developing mouse cortex, and rather than looking at total expression, they looked at relative inclusion of the exon expression, and they did this to look at developing mouse cortex using a time series RNA-seq data, and they're able to identify clusters of exons using the relative expression and identify these sort of temporal clusters of exons going through changes through the developmental program, like either in an early switch pattern or a late switch pattern. So yeah, so as I mentioned, this approach kind of allows you to somewhat look at like an exon skipping network. So going further with exon skipping, if we actually look at the different types of splicing events that occur in different genomes, we can see in the case of humans that exon skipping is the most common form of alternative splicing, but even then it still only represents about half of the possible splice events that we would see in humans. If we go down in the case of mouse, which is what they looked at, we can see that we're even closer now to only representing about 40% of the possible splice events that would be possible to occur. And then if we go in different species all the way down to plants, we can actually see that exon skipping events are actually the least common splicing events in plants. And so if we build a network based on the relative inclusion of exons, we actually wouldn't be representing very much of our genome. If we stick with humans, though, but we address the case of cancer in humans, it's actually well known that we actually have an increase in non-skipped exon events, where we have these sort of cancer-specific splice events. This data is from a study where they looked at the different types of splicing events across different cancer types. And one thing they show is that they have a sort of a decrease in the total ratio of skipped exon events compared to all the other events. And another thing that we've been seeing in cancer is that we actually have an increase in these intron retention events. And so, yeah, just again, like in the case of AML or some of the other cancers, if we were to build a network based on just exon inclusion levels, we'd still only be representing maybe half of the possible splicing events. And this data actually only includes the most basic splicing events. It doesn't include any of the complex events that are actually common, that we commonly see in cancer. And so with that said, if we look at some of these cancer-specific splicing events, we can see we do have exon skipping events, but we do have these other events that aren't just exon skipping, where we might have this exon skipping cluster that it might be significant to a particular drug response in a particular cancer. So if we wanted to do a network approach to look at coordinated splicing, it'd be more beneficial to actually have a network that looks at the expression of these specific variants that are known to happen in cancer, rather than just looking at the relative inclusion of these individual exons. And so this leads me to my second specific aim, which is to develop an event-based splicing network approach to study the co-expression of alternative splicing events and identify and annotate potential splicing network modules associated with drug response in AML. So how I'm going to do this, so if we recall the event-based splicing method where we quantify splicing in the form of these localized splicing events, we quantify these events for all the events that we can find across all of our samples, and then we build a correlation network using the quantified percent splice and other splicing events from our samples. And so to do this, we'd use a similarity measure, just like other co-expression networks. The exact similarity measure that will work for the splicing event case is something that I don't actually know, so this is something that I'll have to evaluate as I work on this project. But essentially what we'll do is then we would have a network where each node of our network represents a particular event from a particular gene, and each edge is a weighted edge representing the correlation between those events based on our sample expression. And then, so in a network analysis, what we want to do is we want to cluster these events to group highly related splicing events and form modules that might be driving our phenotype. And so we can use some sort of clustering approach, and there's many of these clustering approaches. So again, just like the network development, this is something that I'll have to evaluate. So I'd have to evaluate the clustering steps and the different parameters involved. One way we can sort of evaluate how well our clustering data is we can use module statistics. For instance, we can look at intramodule density. We can look at how tightly connected are each node with intermodule, and also intermodule separability statistics, where we can see how separated are the modules from each other as a means of evaluating our clustering procedure and the parameters that we used. So what this does is we actually essentially now form a module network, where we can now look at the relationships between modules and rather just individual nodes. And so one thing that we can do is we can use what's known as the eigenvector of a module, which would basically summarize the expression of the module as a single value. And then we can use this information in basically a new correlation network, where now we can look at the correlations between different modules. And if we see highly correlated modules, it could mean that we may have interacting pathways, but we could also use it to further refine our network and maybe merge our modules. For instance, if they're highly correlated, that may mean that they're poorly defined, and they should actually be merged to make a better module. So if we take a step back and sort of take a high-level view of co-expression networks and why we're clustering data into modules, this is actually a data reduction technique. It alleviates the multiple testing problem. And from a biological standpoint, this makes sense because phenotypes are driven by coordinated changes of genes all interacting within singular pathways. And by doing this, we can now focus in on those interacting genes, or in this case, splicing events, and focus on the pathways that they're involved in and what's driving that particular phenotype. And we can identify the most central components of those particular modules. And in order to find, so after we cluster into modules, we'd want to find significant modules that would be driving our particular phenotype. In this case, it would be drug response for the different inhibitors. So we could, for instance, now include the correlation of drug response with the module eigenvector and look at the trait of interest that way to identify which module is most significant to our trait of interest. Another extension, that's something that I want to do, is also integrate the expression of regulatory genes. So for instance, splicing factor genes are genes that regulate the splicing of hundreds to thousands of other genes. And so we can integrate the total gene expression of these splicing factors. So once we identify significant splicing modules, we can look at what are the splicing factors that are regulating the splicing events driving this particular phenotype. We can also take after identifying a significant module, we can take a more kind of zoomed in approach where we can actually look at the components of that significant module. One way we can identify key splicing events from the splicing module is to look at, for instance, module memberships, the ones that are most highly correlated to the eigenvector. We can also look at what are called hub nodes, so the most centrally located nodes, or the nodes that have the highest intramodular connectivity. These tend to be the most important nodes and maybe the most biologically meaningful for what's driving the overall module. Another approach for looking at keys, in terms of gene networks, in this case splicing networks, where we can identify maybe significant splicing events, is what's known as differential co-expression. And this is where we actually compare networks across different conditions. So if we identify a significant module, and what we could do, and this will depend on the sample size, we could build a consensus network where we identify a significant module, and then we could compare that network into sensitive and resistant across the sensitive samples and the resistant samples and have separate networks and look at changes in the correlations of the nodes. And if we find, for instance, a node that has strong differential connectivity or differential correlations, that may be a potential node of interest. So this has actually been shown to be a means of identifying, like at least in the case of gene networks, identify genes that aren't necessarily differentially expressed, but can still be significant based on differential connectivity. After kind of doing all this, it's important to validate a network. So there's kind of different methods that I'll be looking at in terms of validating my network. One such approach that's commonly used for validating a network is module preservation statistics, where if you build a network based on a particular data set, you try to evaluate it by building another network, an external data set, and looking to see how well your modules are preserved across the data set. Another method is you could use random permutations, where you actually randomly permute the samples in your network and do different random combinations of the expression values. And what you can do here is then you can see that the network statistics that you identify from your network are significant. Like you could create an empirical p-value to show that those particular statistics are actually significant and not something that you would just see by chance. Another way that I'm actually looking at validating it, though, just the overall methodology, is to do an external data set that's known in terms of the splicing network. So I mentioned this exon network of developing mouse cortex. These sort of temporal splicing patterns are actually well known, and it's been well studied in other developmental programs, including the EMT example that I gave earlier. So one thing I'm looking at doing is building my network, which would include skipped exons, but all the other splicing events, is to test it on this data set and see if I can, not get the exact same modules, but see if I could recapitulate some of the same temporal splicing patterns that I would expect to see in a developmental program. There are some considerations of potential pitfalls of this project. One thing that's actually really interesting that I'm going to be looking at is splice events of the same gene. And so one thing to think about is, like, do we treat, I mentioned how a single gene can have multiple splicing events. Do we treat a splicing event that occurs from the same gene? For instance, the two blue events are from one gene, and this green event is a separate gene. Do we treat those events the same as we would for events of the other gene? We might assume that all the events that belong to the same gene might always be highly correlated. We don't know, so that's something I'm going to look at. So a possible solution, if we don't want to bias our network that way, is to maybe remove any of the edges between same gene events. And what we would hope is if they are significant, for instance, in a significant module, if they are highly correlated with another event from another gene, the fact that they're both correlated with the other gene, hopefully that would still stay in the module, and if they're both significant, we would find that in a significant module. Another approach might be to actually aggregate the events to the gene level, where we actually look at the correlation at the gene's perspective based on the overall events. And so this is something that's actually been done for exons, for an exon level gene network, where they aggregated the exon level expression to the gene level. And this might be an option to avoid this bias, is by aggregating the events to the gene level. Another big issue with this data set, though, is the heterogeneity issue, and that we can have we have different artifacts or batch effects. These can be technical artifacts, including RNA quality or library prep, when and where it was sequenced, and also biological artifacts that we would expect to see in a data set like this, where we can have changes in expression or heterogeneity in our expression based on age, gender, the different disease subtypes. There's different subtypes of AML. And another key one here is tissue source. I mentioned earlier that the data is actually derived from either peripheral blood or bone marrow tissue. And we know that we have tissue-specific gene expression and tissue-specific splicing variant expression. So this is something I have to deal with. So for instance, here's an example where we have gene expression that's confounded by these potential artifacts, and it shows that we now have spurious signals in our correlation network. So the idea is to have a method, and this has been looked at. In this case, this is an example of trying to use the principal components, where we identify the principal components that best explain the variability to these confounders and try to regress out that variability without regressing out the signal for the phenotype of interest, and then actually having true signals. So this is something that's really important that I'm going to have to look at for this project. So just to summarize my project as a whole, so the first aim is going to identify independent splicing events for sensitive and resistant AML, while my second aim is going to be able to characterize mechanisms of coordinated splicing promoting therapeutic resistance. But both of these aims, although independent, they're actually highly related, in that both of which will find splicing event signatures of drug response, or potential splicing event signatures. And this may have, hopefully, potential therapeutic implications in terms of aberrant splicing and its role in AML drug response. And also, this splicing event network, I believe, is a new methodology to look at coordinated splicing beyond just cancer. It can look at coordinated splicing in other diseases and other aspects of biology. So with that, I'd like to thank my dissertation advisory committee, the DMI students, faculty, and staff, and an acknowledgement to my NLM training grant. And with that, I will leave it open for questions. Any questions? Rory. Microphone. I can hear you. Yeah. Sure.
Usually what happens in this room is just repeat the question.
Okay.
Okay, so two-part question.
Oh, okay.
The first part is, have you thought about or do you have plans to do any further subgrouping on aspects like the FLT3 ITD or NPM1, for example?
Okay, she's asking if I plan on doing any further subtyping, a subgrouping based on, right, so the FLT3 internal tandem duplication, right. Do you mean in terms of like looking at the splice events separately for the positive and negative? That's actually another thing I might look at doing. So looking at more of like a homogeneous group and seeing how that, if that maybe helps, because that could also affect my results necessarily. So seeing if I only did in the positive case, looked at the splicing events there and kind of seeing how they change across groups. Does that kind of answer your question? Because also, there's also the other subtypes of AML. I think it also kind of would depend on like sample size too.
And then as a follow-up for that, so definitely for some of the drug response, we know that it's related to mutational status.
Right.
So I guess maybe this is more of a comment, but I think if you really so for some of the drugs, there's going to be a really big signal with some of those mutational events, like
right.
inhibitors, for instance.
Absolutely, yeah.
And so just thinking about looking at that co-occurring or that mutation status.
Yeah, actually, I do plan on doing that. Right. Okay. So I didn't. Yeah, definitely. Like for instance, if I can find a significant splicing module, I'd like to see the co-occurrence with the other mutation events. So that's on my to-do list. Yeah.
Yeah, I just think this could be a really nice layer on.
Exactly. Right. That's my plan. Thank you. Oh, Christina.
So you know that you're looking across different similarity measures. What metrics are you actually used to evaluate these?
That's a good question. Ideally, I think she's asking like what are there's different similarity measures, like different correlation measures. So I'll have to sort of evaluate how well those correlation measures work. I think to do that, I'd have to look at different network statistics. I'd have to look at like how like the connectivity of the network, how well preserved. I could maybe do a cross-validation technique to see and do that to kind of assess the overall network structure, and maybe perhaps like split an idea maybe to actually split the data up to see how well, if I pick, for instance, a particular similarity measure, how well that's like represented across multiple data sets. I think, again, data set comes to a size, but exactly how I'm going to assess each measure, I think I don't completely know the answer right now, but that's a huge part of my second aim, though, actually. Shannon?
So in the modeling in the AML paper, mutation, gene fusion, expression, differential expression, co-expression networks were actually looked at. So we now have kind of gene or transcription level components, and the gene fusions in particular are important. So how are you going to, have you thought at all about how to bring it back to those other analyses that were done, or those other?
Yeah, that's a good question.
Right,
there's a lot of things that we do know from that initial analysis. I definitely, and that kind of goes back to what Rory was saying. I'd like to kind of integrate that with the known data to see how well that sort of recapitulates things. In terms of, so you mentioned, like with the co-expression analysis, like. Yeah, right.
You may come away with an interpretation that's being driven by alternative splicing.
Mm-hmm.
In fact, in the FLT3 ITD case, it's not at all.
Mm-hmm.
It has nothing to do with it. So that, I
Right, right.
How are you going to know if it's really driven, what the contribution of splicing is?
That's an interesting point. So I can at least maybe talk about it with like an example. So one of the most mutated genes in the data set was a particular splicing factor, for instance. So it may not be that if that one was highly associated with drug response, it may not be that the splicing events are what are causing drug response. It may be contributing to the change in drug response, but it could go back to the splicing regulator, which is highly mutated, is causing the aberrant regulation of the splicing events. And so really that is kind of the overall kind of goal, is to kind of bring in the splicing layer of it, but I don't necessarily think that's something I'm aware of. I don't necessarily think that the individual splicing events may be the most significant point to it, but it may actually explain some of the mechanisms that are leading to different changes in drug response across the data set. Bob.
The problem with splicing data is that we get a limited amount of money, and so there's a limited number of reads for each of these samples.
Right.
Which means there's going to be a bias in your data to long genes with lots of exons.
Exactly, yeah.
How are you going to deal with that?
That I don't completely know. I hope the splicing event methods can alleviate some of that, but yeah, that is an issue. So the data set, it's relatively small. I did the preliminary analysis to show that it was at least the junctions could be well covered, but that is something I'll have to actually assess the quality. I think one thing I'll kind of have to do, I think it'll be important in the exploratory data analysis to actually look at all the data in the samples and to see kind of what the overall coverage is, to see if there's if I can maybe detect any bias. And actually, yeah, and that's another interesting component, is to actually look at the length of the exons, if I find anything significant, to see if maybe that's a contributing factor to why it's showing up significant and maybe causing some sort of bias in my analysis. But yeah, that's a great point.
That's kind of what I think channels like that. The splicing networks are
Mm-hmm.
going to be smaller than
Right.
the co-expression networks.
Yeah, absolutely.
And so that coordination between the two data sets gets to be difficult.
Mm-hmm.
That's
Guamin.
How do you consider like integrating the results from first aim and the second aim?
Integrating the results.
Yeah, for example, random forest always is divided unit by 10.
Right.
Like the device somehow like significant related response prediction.
Right, and then integrating that with the second aim. Certainly. So one thing I will be doing is I'd look to see if there's overlap with the splicing module. So if there's, and you hope that there would be. If I do find, for instance, a cluster of splicing events, you would hope that those would actually contain some of the significant splicing events that I found. So that's something I would do to see if they're overrepresented in that module.
So from your random forest
Mm-hmm.
classification, you may even try to choose some kind of like threshold in order to collect your significant.
Right. That, I'm not exactly sure what that threshold, how it would do it, but yeah, there would probably be some sort of threshold, or, and then I would also use a holdout validation test. But yeah, that's something I'm thinking about, is what would actually be the threshold for the most important variants that would then represent the signature. That's a good question.
So do you have some kind of alternative approach just in case your random forest?
In case the random forest doesn't work? Well, my initial approach before that was to just do a multiple hypothesis testing approach. So that could be a way, is to actually just go forward and do a differential splicing analysis. I could potentially, though, look into evaluating a different learning model to doing that. But yeah, I do consider that it could end up being that I don't end up using a random forest depending on how things go. Yeah.
So differential splicing analysis is completely different than a random forest.
Sure.
So that would completely change.
Right.
I'm just surprised that was your alternative strategy, because they're completely different.
They are completely different, yeah.
Your aim would completely change. I just think that's something to think about. I think
Mm-hmm.
what we're getting at is, what's your alternative approach? Still trying to meet the objective at the end.
Right, to identify the
Not
signature.
completely shift gears.
No.
Actually, one more question. How are you going to measure your random forest performance?
Outside of the validation test, or?
Because you see you wanted to choose some kind of features from your random forest.
Right, how do I test the,
Yeah, so you can also not only will try to do the business, right?
right,
And you are going to try to generate the random forest for each drug. So you see you get some kind of
right,
hypothesis involved, right?
sure, sure.
And so how you actually try to
Exactly how I'm going to do that, I don't know exactly specifically how I'm going to do that, but yeah, definitely aware that's something I have to do. Any more questions? No? Okay. Thank you.